Next slide. Slide. Good morning. This is Rama, Botany, and welcome to the webinar session. Today we are going to discuss chapter enzymes. Today we are going to discuss the chapter enzymes. And enzymes, what are they? How do they work? And where do they show their effect?
All these are discussed
in this chapter. First of all, brief history about the enzymes. That is discussed under discovery of enzymes. And this discovery was made about 100 years ago only. That means the work was started before 100 years only through an experiment, performing by experiment. And this experiment was by Louis Pasteur. He is a noted scientist in biological field. And this Louis Pasteur, he demonstrated experiment, and by performing these experiments, he found some new things. Like, while performing this experiment, he found that there are certain microbes which plays an important role in this action of enzymes during the discovery of enzymes. And these microbes, they are responsible for one activity, that is fermentation. As we know that word fermentation. What is fermentation? Fermentation means in Telugu pulita antar. That means our mother prepares idli and dosa, like the tiffins. For that preparation, they used this technique, fermentation, while preparing this dough of idli and dosa. They keep the dough for rest. Overnight they soak the material, and that method is nothing but unknowingly they are doing the work, fermentation. So Louis Pasteur also performed the experiment, and during that experiment some microbes, they played an important role in performing the activity fermentation. So what is actually fermentation? Fermentation means this conversion of glucose into aldehydes. That means conversion of glucose into carbon dioxide and alcohol. Therefore, the substance which was involved in that reaction was named as ferments. That means materials or microbes or whatever which helps in fermenting the materials, or which helps in performing the fermentation activity, are called as ferments. Here we are introducing two words. One is fermentation, the other one is ferments, and these two are introduced by the scientist Louis Pasteur. So before 100 years only, the scientists unknowingly they started the works on these enzymes in the process of discovery of enzymes. And while performing these experiments, he discovered one process like fermentation. And for that fermentation, microbes are taking the most important action, or they are playing important role in performing the fermentation, and those are named as ferments. This is the first scientist efforts. Then second, After Louis Pasteur, next scientist is Edward Buchner. He performed the same. That means in the process of discovery of enzymes, Buchner, he separated or isolated the substance from the microbes like yeast. So Edward Buchner, he is isolating the materials or which is responsible for the fermentation process are extracted from the microbes and named them as zymase complex. That means whatever the substance is extracted from the microbes is named as zymase complex. Then next, after Edward Buchner, the third scientist is Buchner. Buchner, he also performed the same process, and he named that material, or he named that process as, or he named that factor which helps responsible for the fermentation process is called as enzyme. So the original word enzyme was coined by Buchner scientists. So here, three scientists, they are playing important role in discovering the enzymes. First scientist is Louis Pasteur, who introduced the process fermentation, and supporting factors or responsible factors are named as ferments. The next second scientist is Edward Buchner, and third scientist is Buchner. He coined the right name enzymes for these materials. Okay, now what are enzymes? How they are? See this. Enzymes are, they are compared with catalysts. We know the word catalyst, which comes in chemistry. And as we know that catalyst is a chemical substance which participates in increasing or decreasing the rate of reaction and gives the end products and they comes out as it is. That means they participates in reaction for getting end products, but they comes along with the end products as it is. That those materials are named as catalysts. So here enzymes, they plays the role of a catalyst, which helps in increasing or decreasing the rate of reactions. So the main important chief character of enzymes is participating, taking part in reactions, and they helps in increasing or decreasing of the reaction. But they do not participate. They do not. They will not be changed in the during the reaction process. Just simply they increases or decreases the rate of reaction. Then next one, enzymes are defined as, what are enzymes? Enzymes are catalysts. It's very clear. That means they participate in increasing or decreasing the rate of reaction, but they do not change. They are not changed during this process. So that is our first at most important character. The next, how can we define these enzymes? Means enzymes are chemical substances which help in enhancing the rate of reaction. That means they may increase or decrease the reaction. Enzymes are defined as chemical substances which help in enhancing the rate of reaction. That means they may increase or decrease the reaction. So here enzymes in further in next classes we will discuss about the composition of enzymes and their structure and their activity. All the things will be discussed in other classes. Meanwhile, we should know that enzymes are chemical substances. They are nothing but chemical substances. And these chemical substances, who plays the role of catalyst, they helps in increasing, that means enhancement of the reaction. That means that they may increase the reaction or they may decrease the reaction. Then, in other words, enzymes are also be defined as chemical substances which alter the rate of reaction, which alter the rate of reaction. That means they are changing the rate of reaction. They are changing, either decreasing or increasing. By the influence of this catalyst, by the influence of these enzymes, reactions may, rate of reaction may be decreased, or rate of reaction may be increased. So there is a fluctuation by the influence of these enzymes. So enzymes are also be defined as the chemical substances which alter the rate of reaction. So these are the few important general characters of enzymes. And here we can see the uh, structure of enzyme. Here we can see the structure of enzymes. See here the diagram is there, in which enzyme is having some notches. Pencil. Pointer. Sir. Guide ఉంది కదా? Just. So here we can see the diagram of enzyme, and structurally it consists some notches. Okay, sorry for the interruption. Here you can see the structure of enzyme, where you'll find some notches, and this notch is called as active site. Here the reactions takes place after receiving a substrate molecule. See here substrate molecule, and here is active site. So in the place of active site, this substrate molecule will be induced into it, and after that they will make as a complex, and reaction takes place between this enzyme and substrate molecules, and the end product will be given out. So this is about the general points about the enzymes. Then next, here look at the diagrams, how the enzyme will be and how the substrate molecules are, and how the enzymes connects with the substrate molecules, and how they will give you end products, and the enzyme will come out as it is without any without undergoing any changes. So all these are very clearly observed in this slide. So enzymes, usually they are also chemical substances. By having this active sites, observe the shapes of substrate molecules and active sites. Both are similar in shapes. So there is a resemblance between these active sites and substrate molecules. So similar shape of substrate molecules reaches the similar sites, and they fit in the enzyme active sites. And after inducing these substrate molecules into the active site, they develops as enzyme substrate complex. So this is a temporary state. Enzyme substrate complex is a temporary state, and after or furtherly that shows some changes by undergoing chemical reactions. And after completing the chemical reactions, the substrate molecules, they comes out in the form of end products and enzyme that comes as it is without undergoing any changes. So generally this is the nature of enzymes which are shown in this slide. The next, yes, how much rate of reaction can be changed in the presence of enzymes? Just now only we defined the enzymes as the chemical substances which plays the role of catalyst, and they helps in increasing or decreasing of a reaction, rate of reaction. Then no doubt they helps in altering the rate of reaction. Then how much rate of reaction can be changed in the presence of enzymes? Is that more than the absence of enzyme, or is that less than the absence of enzyme? If we discuss this point, say for example, this can be explained by taking one example. Here one equation is there, that is CO2 plus H2O. CO2 plus H2O, they undergoes chemical reaction and gives the end product as bicarbonate, that is H3CO3. This is a reaction. By taking this reaction, we can understand how much of the rate is changed by the influence of enzyme. First, this reaction is taking place in absence of enzyme. Then what is the end product, rate of end product, or what is the rate of reaction? See, the rate of reaction in absence of enzyme is 200 molecules per hour. Per 1 hour, here equation is there. CO2 plus H2O gives rise H3CO3. And for this, for getting this end product, there is no need of an enzyme. That means enzyme is absent. Enzyme is not taking part in this chemical reaction. And what is the output? Output is 200 molecules per hour. It's very clear. That means without enzyme influence, the end product is 200 molecules per hour. Right. The next step. Now the same reaction will be observed in presence of enzyme, and how the rate will be changed. Look at this reaction. The same reaction. First we observed without the influence of enzyme and observe the rate of end product. And here with the influence of enzyme, the rate of reaction, how it will be influenced by the enzyme. See here, the end product rate is, and here the enzyme is carbonic anhydrase. Here enzyme is involving in this reaction, and that enzyme is carbonic anhydrase. And the end product rate is 6 lakhs per second. Listen it carefully. 6 lakhs per second. That means a lot of change is there in rate of reaction. Without the enzyme participation, rate of a reaction is only 200 molecules per hour, whereas by the influence of an enzyme, or here especially carbonic anhydrase, with the influence of this enzyme, the rate of reaction is enormously increased. That is 6 lakhs per second. So here we can understand how the enzyme shows its effect in increasing the rate of reaction. So here this reaction takes place in the presence of enzyme like carbonic anhydrase, and this carbonic anhydrase is the fastest enzyme. And here the product is same, H3CO3 only. Product is same, substrate molecules are same, but this reaction can be handled in two ways to understand the fluctuation in rate of reaction in the organisms. So here we can understand the enzyme influence. We can understand the influence of enzyme on the chemical reactions. So by the influence of enzyme, rate of reaction is increased very much. It is that means enzyme is showing a lot of effect in increasing or decreasing the rate of reaction. But here we are observing only the increase. Here without enzyme it is resulting only 200 molecules per hour. But whereas by the entry of this enzyme, the same reaction will give you a large number of molecules of end products per second. Time is also be reduced and number of molecules are increased by the influence of enzyme. So the number of molecules produced are 6 lakhs per second. By this we can understand the rate of reaction is many times higher in presence of enzymes like carbonic anhydrase. And this carbonic anhydrase, originally it is the fastest enzyme that works very speedily. So in the presence of this fastest enzyme, a rate with which it enhances the rate of reaction is known as turnover number. What is turnover number? That means with the influence of this enzyme, like carbonic anhydrase, which is the fastest enzyme, fastest activating enzyme, and in the presence of that enzyme, rate of reaction that is enhancing. That means rate with which it enhances, that rate is called as turnover number. What is turnover number? It comes under 1 mark in multiple choice questions. So try to remember this point, turnover number. That means enhancing number with which it enhances the rate of reaction in the influence of enzyme is called as turnover number. So finally, what can we say? So we what exactly the enzyme is? What is enzyme? Means it is the helping. Enzyme is helping. It is helping in altering the reaction. What is the enzyme? Enzyme can be defined as like this. Enzyme is helping factor which helps in altering the reaction, rate of reaction, or changing the rate of reaction. So how much rate of reaction is changed in the presence of enzymes can be understood by this example. So without enzyme influence, rate of reaction is minimum, but by the influence of enzyme, rate of reaction is changed enormously, or the rate of reaction is changed very high or many times higher. So that can be understood by this example. Next. Next slide. Next slide. What are the characters or chemical nature of enzymes? Before discussing about the nomenclature, we should know the chemical nature of enzymes. What are the properties of enzymes? How they are framed, and what are their important factors? See, most of the enzymes are proteins. There is a statement. All the proteins are, all the enzymes are proteins, but not all the proteins are not enzymes. That indicates that the majority of the enzymes are made by proteins only, but all the proteins are not enzymes. And tertiarily, the specific, then some of them work without any association with proteins. That means by nature they are proteins, and some of them work without any without anything associated with the proteins. That means with the association or without the association of other things, these proteins are showing their effect. Then next, on the basis of this, enzymes are classified into two types. That means first, what is the chemical nature means? Naturally, they are proteinaceous. That means enzymes are made by proteins, but all the proteins are not enzymes. Then these proteins, they may show their effect by the association of other non-proteins or association of the proteins. That means they may work with the association of any other thing or may not. So based on this factor, these enzymes are classified into two types. One is simple enzymes, the another one is conjugated enzymes. The next, first let us discuss about the simple enzymes. What is simple enzyme? That means when the protein is acting as enzyme without any non-protein part associated with it is called as simple protein. Just now only we discussed that all the enzymes are proteins, but all the proteins are not enzymes. So definitely we should remember this point. Why? Because enzymes are made by only proteins, but not any other. And here, this protein-made enzymes, sometimes they associates with another non-protein part to show their effect on the reactions. But at the same time, they may not associate with any other part. Then this non-associated protein type of enzymes are called as simple enzymes. So here the definition is protein is acting as enzyme without any association, without any non-protein part association. That means only protein part that plays a role of enzyme. Only that protein acts as protein without any association of non-protein part. Then that type of enzymes are called as simple enzymes. The next, for this example is pepsin and trypsin, which are observed during the digestive system. So all the majority of the digestive enzymes, they comes under simple enzymes only. Why? Because only protein part that acts as enzyme without associating with any other non-protein part. The next, these are the enzymes which do not require anything to come and bind with the protein part. That means these enzymes, they do not require any non-protein part to bind with the substrate molecules. So this protein part is self and is working as enzyme. What we have to understand here is only for protein part that acts the role of enzyme without the association of any other non-protein part. So simply protein plays the role of enzymes and those are simple. That's why they are treated as simple enzymes which are observed during the digestive system. And in that digestive system, pepsin and trypsin like enzymes, they comes under the simple enzymes category. The next one is conjugated enzymes. Conjugated enzymes. Most of the cases there is a protein molecule with an association of non-protein molecule, and they helps in binding the substrate molecules attached to that. By that, this protein molecule becomes functional, and this type of enzymes are called as conjugated enzymes. Here we have to notice one thing. In addition to protein molecule, there is definitely one non-protein molecule. Then only this enzyme will be activated by the association of non-protein molecule. Then only it becomes functional. Then only it shows or it participates in the reaction and gives the end products. So such type of enzymes are called as conjugated enzymes. So try to differentiate a simple and conjugated enzymes. In simple, only protein molecule that plays the role of enzymes without the association of non-protein part. Whereas in conjugated enzymes, along with the protein particle or along with the protein molecule, definitely there should be a non-protein molecule, and by that association of non-protein molecule, then only this enzyme that becomes activated or that will be activated and that becomes functional. And this functional enzyme that takes part in the reaction, that takes part in the reaction. So this type of enzyme is called as conjugated enzymes. So only the difference between the simple enzymes and conjugated enzymes is, in conjugated enzymes, definitely the protein molecule associates with non-protein molecule to make it functional. That's why it is called as conjugated enzymes. And when the protein molecule is not associates, or when the protein molecule do not associates with the non-protein molecule, then that comes under the simple enzymes. So these are the two important types of enzymes. Then, and conjugated enzymes, and this protein part, that means the basic protein part is named by apoenzyme. Protein part is named by apoenzyme, and non-protein part is named as cofactor. So in conjugated enzymes, protein part is named as apoenzyme, and non-protein part is named as cofactor. So definitely we should remember these 2 points. In conjugated enzymes, protein part is called as apoenzyme, non-protein part is called as cofactor. Then in simple enzymes, there is no question of apoenzyme, and a cofactor. Simply they are called as simple enzymes. But whereas in conjugated enzymes, we are defining these 2 parts. That means the protein part and non-protein part. They are named differently. Protein part is named as apoenzyme, and non-protein part is named as cofactor. So, and protein part on its own is non-functional. That means in conjugated enzymes, the only protein part, it is functionless, it is inactive, it is non-functional. By the association of a non-protein, it turns into functional. That is an important point which should be remembered during this conjugated enzymes. In conjugated enzymes, 2 parts are there. One is protein part, another one is non-protein part. This protein part is naturally a protein, which is called as apoenzyme, and non-protein is cofactor. In this conjugated enzyme, this apoenzyme remains non-functional or inactive or functionless. ఎప్పటివరకు means by the association of non-protein molecule వరకు, అంటే non-functional protein molecule అనేది associate అయ్యే వరకు కూడా అది non-functional. That's why this APO enzyme remains non-functional until and unless the non-protein molecule comes and join with this APO enzyme. Appadi varaku kuda that remains as non-functional. So this non-protein molecule is called as cofactor. Okay, why it is calling as cofactor means this is a factor. Which one? APO enzyme is a factor, and here cofactor, but co means associate. uh, which molecule is associating with the factor? That means non protein molecule is associating with the protein molecule. That's why this non protein molecule is considered as cofactor. that's uh, that's the reason of giving name cofactor. The next, after the association of the protein and a cofactor, that means non-protein molecules, the resultant enzyme is called as holoenzyme. That means here conjugated enzymes, totally they are named as holoenzyme. That means one protein molecule, it is associating with the non-protein molecule, and after the association with the non-protein molecule, this whole conjugated enzyme is called as holoenzyme. So what is holoenzyme? That means association of a protein with the cofactor, resultant enzyme is called as holoenzyme. So here we should remember 3 points. One is simple enzyme, that is nothing but protein, only protein part that takes part in the in altering the rate of reaction. That enzymes are called simple enzymes. And in conjugated enzymes, along with the protein molecule, there should be the definite non-protein molecule. Then only those are called as conjugated enzymes. In conjugated enzymes, protein part remains inactive or functionless. After the association of cofactor or non-protein molecule, then only it becomes active, or then only it becomes functional. That's why this non-protein molecule is treated as cofactor. So finally we can define what is conjugated enzymes, or is there any other name for after joining with the non-protein names? Yes, there is other name after the joining with the non-protein molecule is holoenzyme. What is holoenzyme? That means association of protein with cofactor or non-protein molecule, which results in the formation of an active enzyme, which is called as holoenzyme. So these are the chemical nature of enzymes. Next slide. Right. Here, some more details about the cofactors. Usually these cofactors, they are non-proteinaceous. And these cofactors are classified into 2 types. One, organic non-protein substances, and second is inorganic metallic ions. And this organic non-protein substances, again furtherly they are classified into 2 types. One is coenzymes and the other one is prosthetic group. On what basis these organic non-proteins are classified? Means this non-protein substances, they associates with the protein molecules, isn't it? So there is an association of protein with the non-protein substances, and this association may be weak or strong. That means as per the requirement, this association may be separated, or it remains remains permanently, or they cannot be separated. So based on that factor, that means on the basis of a separation from protein molecule. Try to understand the point. Here, cofactors are classified into 2 types. Primarily, we should know the meaning of cofactors. Cofactors are non-proteinous or non-proteinaceous substances. These non-proteinaceous substances are classified into two types. One is organic and the other one is inorganic types. In under organic non-protein substances, again there are two types: coenzymes and prosthetic groups. These two types are classified on the basis of the separation from protein part. So here, if the non-protein part separates or cofactor, if it separates from the protein part very easily, then they comes under coenzymes category. And if a non-protein particle or non-protein substance or cofactor, if it cannot be separated from the protein part, then that comes under prosthetic group. That means easily, if the non-protein group is separated from the protein part, then that comes under coenzymes. And if it is not separated, then that comes under non-prosthetic group. So based on this separation factor, separation of non-proteinaceous group, these are classified into coenzymes and a prosthetic group. The next one, under inorganic metallic ions, there are no other groups. So here, these cofactors, they are classified. If they are separated easily from the protein particles, then they comes under coenzymes, and if they cannot be separated from the protein group, they comes under prosthetic group. This is clear. The next, on the basis of working site, that means site of enzyme action. Just now only we have seen and the hello. Yes. Just now only we have observed the structure of enzyme site. Here, this is the enzyme molecule, and this enzyme molecule is having some notches, and this notch is called as active site. And active site is the place where the substrate molecule comes and fit in this site. That means inducing of a substrate molecule takes place in the active site. So this is the general structure of an enzyme. Okay, right. This is the structure of enzyme. The next, here, on the basis of working site, these enzymes are classified into two types. See here, just only I have shown you the active sites or enzyme sites. They are the functional sites of the enzymes, and on the basis of that working site, these enzymes are classified into two types. One is endogenous or intracellular enzymes. Intra means within the cell. Within the cell, if the enzymes are produced and if they works inside the cell, then those type of enzymes are called as endogenous or intracellular enzymes. The next, exogenous or intercellular enzymes. Inter means in between the cells. That means for this, the best example is digestive enzymes. If we take this bowl, what is that? Not stomach or stomach. You take better the stomach. Stomach is a sac-like structure in which the food, whatever we consume, that goes and settles in that stomach, and there it remains for 2 to 3 hours. And during this duration, various enzymes are secreted by the cells of this stomach, and they mix with the food particles and undergoes the digestion process. That means here the reactions are taking place outside of the cell. That means the space which is outside of the cell. So that means in the lumen, which is called as lumen. So in that lumen, the changes are taking place by the secretion of the cells which are situated in the walls of the stomach or alimentary canal. What we have to understand here means the enzymes, they are secreted by the cells into the open place which is called as lumen. So here enzymes, they are showing their effect. They are working outside of the cells. That's why this type of enzymes are called as exogenous enzymes or intercellular enzymes. For this, the best example is digestive enzymes, which are secreted by various cells, acts on food and the lumen of alimentary canal. That means it is taking place outside of the cells, right? That means the enzymes, they are working outside of the cells. That's why they are called as exogenous enzymes. But there is an exceptional case, that is in unicellular organisms. It is intracellular type of enzymes. In unicellular, the whole organism is with single cell. Whole systems are arranged in that cells only. That's why in this unicellular organisms, enzyme action is intracellular type of enzymes are found in this unicellular organisms. So today we'll stop it here, and in the next will be discussed in the next class. Thank you.
Again,